All right. I always feel like we've reached the pinnacle of a mountain when we finally get to this part of the course, because we started a rather long journey. Looking, of course, at Gregor Mendel crossbreeding pea plants and trying to solve the problem of heredity. And every new thing that we found raised more questions, as good science should. And finally, we've gotten through to a point where we can start really seeing how everything works. You can see a lot from the top of this mountain that we have just reached. So what does this mountain look like? Let's recap a little bit. Organisms have the traits that they do because they inherit these things called genes. This is what Mendel basically worked out. Mendel did not ever get to find out what genes really were, but they behaved like particles, and he called them elements again. And this is fundamentally what heredity is about. We talked about heredity way back in the beginning as the way in which organisms come to resemble their parents, whether plants, people, or protists, whatever it is that you're working with. Now, about the time that Mendel published his work, microscopists, various people who were specialists in the use of the microscope, were beginning to work out that when a cell divides, it shows these thread-like things appearing in its nucleus, and we call those chromosomes. And when a cell divides, these move in a series of steps, a little dance that they do called mitosis. However, when cells go on to become gametes, that means eggs or sperm. Could somebody mute? You're getting some static and some scratching noises coming in. Somebody could please mute. Organisms that do that undergo a different type of division, and that reduces their chromosome number by half. In your normal cells, you've got 46 chromosomes, but your eggs or sperm, depending, only carry 23. And that type of reduction division that, at least in animals, including people, is only seen in the production of gametes, we call that meiosis. Now, at first, it wasn't clear what the connection was between these, but Thomas Hunt Morgan and his students clinched the case that genes are physically located on chromosomes, that what genes fundamentally are is they are parts of chromosomes. Chromosome can be thought of as a long strand of genes. Okay, great. Well, we know that chromosomes are long pieces of DNA, very long strips. If we took all of the DNA out of one of your cells and lined up all of the chromosomes end to end, we'd get about 6 feet of DNA divided into 46 chromosomes. When chromosomes undergo mitosis, they're wrapped around proteins called histones, but histones don't do the work. DNA does. Could people please mute? Sorry, somebody's got a lot of random background noises coming in, and it's honestly kind of throwing me off my game a little bit. Well, DNA, as Watson and Crick and Ross Franklin worked out, is a double polymer of nucleotides. Remember, a polymer is any kind of molecule that consists of a bunch of subunits strung together in a very long line. So DNA is a double polymer, two polymers wrapped around each other in the famous double helix shape. What does DNA do? It stores information, and the information is coded in the order that the nucleotides are in. This is the so-called genetic code. Just like the order of letters is important for carrying information in a word or a sentence, the order those nucleotides go in is important for carrying information that gets passed on from one generation to the next. So DNA stores information in that nucleotide order. That information, yes, there is a way I can mute people, but I can't easily reach it while at the same time showing slides is the thing. That's why I've got to ask you to please mute. That information is copied from DNA onto a piece of RNA, and we call that copying process transcription. The RNA is read at a ribosome, one of, okay, all right, it's okay that you forgot to mute your mic. Just keep an eye on it, but thanks. RNA is read at one of many, many thousands of tiny structures inside a cell called a ribosome. And the information carried by that RNA is used to assemble amino acids into the right order into proteins, and that's a process called translation. Now, new concept, that information flow stored in the DNA, copied by transcription onto a piece of RNA, and then used to assemble a protein by the process of translation. We call that the central dogma of molecular biology. Information flows only one way in a cell. The proteins cannot come back and rewrite your DNA. They can do some interesting things with it that we just have not got the time to get into. If you really want to know, you can major in this stuff. But information flow in a cell only goes in one direction, and we call that the central dogma. And like all dogmas, it's got a really interesting exception to it, but I'll get to that in just a minute. So that's a recap of how we got to this mountain peak where we are. All of those things that I've touched on, by the way, are things that you really need to know well on the second exam. You know, be able to parse the difference between DNA and RNA, the differences between mitosis and meiosis, what goes on in transcription and translation, and so on. All right, be able to retrace our steps up that mountain. Everything I've talked about fits together as a step on that journey. One of the things that we can see from the top of our mountain is evidently some discolored plants, which is perhaps not the view you were looking for. These are tobacco leaves, and tobacco farmers knew about a disease that their crop could get. Plants that were affected by this disease developed irregular patches on the leaves of discoloration. You can see how these leaves aren't solid dark green. They've got kind of yellow-green patches on them. And the disease was called tobacco mosaic disease. Remember, a mosaic is a picture made by putting pieces of tile together to make a pattern. And these leaves look like, you know, you have patches of green and yellow that have been put together, hence the name. Now, scientists were looking for, in the late 1800s, were looking for ways to stop tobacco mosaic disease. Tobacco was an important crop. And they were looking for bacteria that might be causing the infection. Bacteria can infect plants the way that they can infect animals. There are bacterial diseases that your plants can get, but they couldn't find any bacterium that was causing tobacco mosaic disease. All right, just checking the chat. A Russian guy named Dmitry Ivanovsky, and independently of him, a Dutchman named Martinus Beijerinck, who's shown here. This is Beijerinck, working independently of each other. This was a time when communication internationally was a lot slower and more difficult. They found that you could squeeze out the juice of an infected leaf and smear it on a healthy plant, and the healthy plant would get the disease. But they tried filtering the juice through very fine porcelain filters, filters with such tiny holes that every known bacterium was stopped, and even the filtered juice would cause the disease. So there had to be something in that juice of the infected leaves that was smaller than any bacterium, smaller than any light microscope could see. And yet whatever it was must be able to cause the disease in a healthy plant. So there's something that's acting like an infectious bacterium, but it can't be a bacterium because it's way too small. Beijerinck borrowed a Latin word that originally meant poison and named whatever this unknown thing was that was causing tobacco mosaic disease a filterable virus, which we typically shorten now to just plain old virus. I know you've heard a lot about viruses, especially you've had a belly full, not literally, I hope, of the coronavirus. There's lots of other viruses out there, and I haven't talked about them before now because to know what viruses do, you do have to get some pretty good background knowledge in molecular biology. But now we can finally introduce what viruses are and how they work. And this is how they were discovered. It didn't become possible to see what viruses were until the 1930s, when a much more powerful type of microscope was invented. You don't use this in class because the scopes that you use, the light microscopes, cost a few $100s. A typical electron microscope runs into 6 figures. So we don't really have, and takes up a small room. So we don't really have personal electron microscopes for you to use. But electron microscopy allowed us to see the tobacco mosaic virus for the first time. And this is what it looks like. It's not made of cells. It looks like very long rods, but it's not a bacillus. It doesn't have a cell membrane or a cell wall. It's not a cell at all. It's only, remember that a micron is 1/1,000,000th of a meter. And this thing is 3/10ths of a micron long. That's 10 times smaller than a typical bacterium would be. And what's more, it's completely inert. If you take tobacco mosaic virus out of a tobacco plant, it'll just sit there. It has no metabolism. It doesn't eat. It doesn't excrete. It doesn't reproduce. It's got no more metabolism than a rock. It does absolutely nothing. And in fact, you could make crystals out of it. So what is this thing that causes disease and yet isn't a cell and doesn't have metabolism? Well, it's a virus. Viruses turn out to be pieces of either DNA or RNA. And that DNA or RNA is surrounded by a coat of proteins that fit together. And that coat is called a capsid. Viruses are virtually all too small to see with a light microscope. Some of the largest ones you might be able to pick out under a really good light microscope, but it's still just going to look like a dot. They are smaller than most bacteria. Most of them you can't see with light at all. They're not cells, and they're not made up of cells. And when we talked about all those features that living things have way back in the beginning of the course, viruses kind of straddle the boundary between living and non-living because they have some of those features, but they haven't got all of them. So the question as to whether you should call viruses truly alive or not alive is a really good topic for late night coffee fueled discussions up in your dorm room, if students still do that. When you isolate viruses, they just sit there. They are completely inert. As I said, if you squeeze a bunch of tobacco mosaic virus out of infected plants, you can keep it in a test tube on your desk and the virus will do nothing. It won't metabolize, it doesn't eat, it doesn't poop, it doesn't breathe, it doesn't reproduce, it just sits there. It's got about as much life in it as this bench that I'm sitting at. But when you bring viruses close to a host cell, viruses will inject their DNA or their RNA into the host, and a virus-infected cell begins duplicating the virus's DNA or RNA, and it begins transcribing and translating the virus genes to make virus proteins, and it turns the cell into a factory that, instead of making the proteins that that cell needs, makes more viruses. And the viruses eventually burst out of that cell and go on to infect other cells. You might remember computer viruses. Computer virus is a program that, if it enters your computer, it takes over part of your computer's function and turns the computer over to making more copies of itself and spreading them to everybody on your email list or what have you. Computer viruses are analogous in this way to biological viruses. A computer virus doesn't do anything on its own. It's just a little short piece of code. But when you introduce it into a computer, it turns the computer into a factory for sending out more copies of itself. Viruses do much the same. They turn a host cell into a factory to make more viruses. It would be a little bit like if, well, never mind, I'm not going to go there. That's a picture of the outside of one virus in particular, and this happens to be Zika. The Zika virus, you might remember from what, maybe 7 years ago, as a virus that's transmitted by mosquitoes and that had the potential to cause birth defects in the fetuses of pregnant women who got infected. This happens to be what the capsid looks like, and it's actually rather pretty. Each one of those different colored regions is a different protein. You have little blue spike proteins and then red, orange, and yellow capsid proteins that pack together in a symmetrical way. And you can see there's actually, quite a bit of symmetry here. You've got the red and the orange make these little three-armed figures that look like the plastic you would put in the center of a 45 RPM record, except none of you know what those are. And the yellow kind of, looks like stars and things like that. This is rather attractive. So that's an example of a capsid. There's the capsid of tobacco mosaic virus. The proteins are shown in yellow, and they pack together tightly to make that long cylinder that you saw earlier. And the RNA, which is shown in red, is coiled up inside the capsid. And by the way, this was the other major accomplishment that Roz Franklin did. Roz Franklin was able to crystallize tobacco mosaic virus and work out the basics of its structure using X-ray crystallography. That's basically how this picture was made, by the way. We've gotten quite a bit better at it, and this is how we can reconstruct what viruses look like in great detail, but it's still basically X-ray crystallography, the same technique that Roz Franklin used. And that was the other big thing that she did. Okay, so the capsids on the outside and DNA or RNA, depending on the virus, is coiled up inside. This happens to be a virus that infects bacteria called the T4 bacteriophage. And incidentally, I know somebody with bacterial pneumonia, a lung infection that's resistant to antibiotics because of, natural selection, who's getting an experimental treatment with bacteriophage. They're sending in these viruses with the hope is that they'll kill the bacteria in her lungs, but they'll leave her own cells alone because most viruses can only infect a very limited number of cell types. And bacteriophage cannot infect human cells. So anyway, there's a computer reconstruction of what it looks like on the right. The DNA is all coiled up in that head that's shaped kind of like a 20-sided die. There's a stalk coming down from the head, and then these little fibers sticking out, and those are all different proteins. And on the left, there's an electron microscope photo of the T4 bacteriophage. Okay, here's another electron micrograph. This is a picture of a bacteriophage virus that has latched onto a host bacterium and is inserting its DNA, in this case, into the host. There's that 20-sided die-shaped head, and there's that shaft with the tail plate, and there's those fibers that the thing is using to attach to the host cell. And its DNA is located up in here, and that DNA is getting injected into the host bacterium. That host bacterium is getting a shot of phage DNA. And what happens now is that the bacterium begins to transcribe and translate that phage DNA, and the bacterium begins to replicate that phage DNA. And the bacterium becomes a factory in which phage DNA and phage proteins get self-assembled into new virus particles. The capsid proteins self-assemble into new capsids. Copies of the virus DNA end up inside the capsids. And eventually all these new viruses come bursting out of the host cell, which generally kills it. So this is a bacterium caught in the act of exploding, and you can see a whole bunch of these phages bursting out of this unfortunate bacterium. And inside the bacterium, you can see a whole bunch more capsids, a whole bunch more virus particles that are in the process of getting assembled. Incidentally, there are some viruses that can screen themselves from the host's immune system because they take pieces of the host cell membrane with them. These are pictures of a human herpes virus. Herpes viruses, one type of them causes genital herpes. A different type of herpes virus causes cold sores. And in these images that you're seeing right here, the capsid is right there. I'm circling them in red. But when herpes viruses come bursting out of the host human cell, they're wrapped in a little piece of the host cell membrane. And that's what this thing is that I'm circling in red right now. So some viruses do that. And this is your friend and mine, the goddamn coronavirus. Now, coronaviruses have a capsid. Coronaviruses wrap themselves in a cell membrane like the herpes virus does. But on the outside of that cell membrane, you have spike proteins that are sticking out. And I'm drawing them in red up at the top. Down at the bottom, that's a computer graphic model of a coronavirus. And you can see the spike proteins sticking out because they're in red. And the spike proteins resemble a crown, which in Latin is corona. That's why coronaviruses are called that, because they look a little, if you're on drugs, like crowns under an electron microscope. And those spike proteins are also called fusion proteins. If you've been keeping up with medical advances, you might know that the vaccines target the spike proteins, because the spike proteins are what coronaviruses use to recognize and attach to host cells. So there. That's a section through an infected human cell that's been infected with coronavirus, and every one of these little circles that you're seeing is a new coronavirus particle being made inside the host cell. So this is what all viruses do. If you get coronavirus, the virus particles enter your lungs. They attach to cells in your lungs, and they turn your lung cells into factories for making more coronaviruses. The coronavirus takes over a cell's machinery and use it to make more copies of the virus. And then those come bursting out and they spread to other cells in your lungs and so on. Here's why all of that crap I was talking about with protein shape is necessary. Cells have proteins on their surfaces, and you can think of them as being like antennas that enable cells to sense and respond to their environments. In multicelled organisms, including humans, some of these cell surface proteins respond to messages that the body sends out called hormones. The reason why you went through a stage when you grew really fast and was always hungry and looked like you were made out of knees and elbows is because you started making certain hormones when you reached puberty, and cells all over your body had these proteins on them that responded to the hormones, the chemicals that your ovaries or testes were sending out, and caused you to, grow really fast and get really moody and have crushes on people and act really stupid. This is a human cell receptor protein. It's called ACE2. And it enables the cell to respond to the hormone that regulates blood pressure, a hormone called angiotensin. And part of this protein is outside the cell and part of it is inside. The way this has been drawn, all of these things that look like rods are actually alpha helices. So it's a fairly complicated protein. And what you're looking at here is in blue, that's a host cell surface. That's what the surface of one of your lung cells would look like if you could see it really highly magnified. And those blue things are the ACE2 proteins on the surface of a host cell. The spike proteins of COVID, which I'm outlining now, which are shown in this kind of salmon pink, they bind to the cell's ACE2 proteins, and that's what allows the virus to enter. If the virus didn't make spike proteins of that shape, the virus would not be able to latch onto ACE2 proteins and they wouldn't be able to get into the cell. Sorry, that hopefully that cough is just nothing. That's why all of that stuff we were talking about protein shape matters. COVID is infective because the COVID virus has spike proteins of just the right shape to latch onto ACE2 proteins in your cells, on the surface of your cells. By the way, your body defends against viruses using a population of white blood cells that are called cytotoxic T cells, or as they're also called CD8 cells. This is a colorized picture. This is not the natural colors. But in orange, that thing is a cytotoxic T cell. And the cytotoxic T cell is buddying up to, in this case, a cancer cell, which is shown in purple, but it would also buddy up to a virus-infected cell in the same way. And a cytotoxic T cell destroys virus-infected cells before they can release the viruses. Now, of course, it kills off the cell, but it kills off the cell before it can release these viruses. This is the way that you defend against virus infections. And again, CD8 cells also defend against cancer. Here's the thing, the human immunodeficiency virus, or HIV, is another virus that wraps itself in cell membrane. You're looking at an HIV-infected cell. There's lots of virus particles down here, all of these little black specks. And then as it buds off the host cell, it wraps itself in a bit of cell membrane. So that right there is a mature human immunodeficiency virus. And that's another one, and that's another one. And you can see some that are budding off. And what makes HIV dangerous is it attacks CD4 cells, which help your CD8 cells. So what makes HIV dangerous is it damages the body's ability to defend against viruses. You know, it's a virus that attacks your virus defense, like an like an anti-missile missile. And HIV is an example of what we call a retrovirus. Retroviruses are called that because they're always seen wearing bell-bottom jeans and they've got lava lamps in their house. Actually, I am kidding. Retroviruses contain RNA, not DNA, but they also include an enzyme called reverse transcriptase, which takes the virus's RNA and makes a complementary DNA strand. And that new DNA gets spliced into the host DNA and is transcribed and translated to make new retroviruses. That is the only major exception to the central dogma of molecular biology. Almost all the time, DNA is stores information, information's copied onto RNA, and then used to assemble proteins. This is one of the few cases where information is stored on RNA and copied onto a piece of DNA. Retroviruses do that. They're the primary exception to the central dogma of molecular biology. There are some other viruses that do this. The first retrovirus to be discovered, if memory serves, actually causes a type of tumor in chickens. It's called the Rous sarcoma virus. HIV is by far the most famous retrovirus that's out there. There's another one called HTLV that causes a form of leukemia. If you have cats, you might know about FIV, feline immunodeficiency virus, which damages your cat's immune system. Those are retroviruses, the main exception to that central dogma. Antibiotics, by the way, do absolutely nothing against viruses. Antibiotics block bacterial metabolism. Viruses don't have any metabolism. Viruses don't eat. Viruses don't grow on their own. Viruses don't do anything that antibiotics are effective against. Incidentally, pretty much the same is true for, ivermectin, which is a very effective drug against worms and other parasites, but the clinical trials that I've seen show that it really doesn't do anything against viruses. There is a drug you can take called interferon. That's a protein, and it's shown here, and you can see it's got lots of alpha helices. And we don't have time to go into exactly how it works, but it stimulates the immune system to step up its game. But here's another antiviral drug. Up at the top, that's a real nucleotide. And for the exam, you need to know nucleotide structure really well. You don't have to draw these formulas, but you need to know the parts. Up there at the top, I've got a phosphate group, and that's attached to a simple sugar. In this case, it's deoxyribose. And that sugar is attached to a base, and that's a single or double ring of carbons and nitrogens. And in this particular case, the base is guanine. So up top, that is the nucleotide, guanidine phosphate. You've got lots of that in your own DNA. It's one of the basic four. Right here, down below, we've got guanine, but it's not stuck to ribose. It's just stuck to a couple of carbons instead of a complete ribose sugar. This is a fake nucleotide. This looks like a nucleotide, and if you have a virus infection, the cell will take these fake nucleotides to try to make virus DNA or virus RNA, but it's just different enough that it can't actually be used. If a cell tries to use these in DNA replication or in transcription, the process is shut down. The process gets blocked. And this fake nucleotide happens to be the drug acyclovir, which is used as a treatment for herpes virus. The parts of a nucleotide, we've got the phosphate up here. That's a phosphorus atom with a couple of oxygens. The sugar down here, and in this case, it happens to be the sugar deoxyribose. That's a ring of carbons with a few oxygens. And the base over here is either a single or double ring of carbons with lots of nitrogens as well. There's the parts of a nucleotide. Here's another nucleotide up at the top. There's your phosphate right there. Your sugar, that's deoxyribose. And a double ring of carbons and nitrogens. In this case, this is adenine. Down here, we've got a fake nucleotide that has adenine, but instead of being the normal sugar, it's a sugar that's got this weird nitrogen stuck onto it. The name for this is galidesivir. It looks like it's effective against several virus infections, and I'm not sure of the results, but it was put into clinical trials for effectiveness against coronavirus. Here's another one. This is yet another nucleotide. Here's adenine. There's that phosphate right there, the sugar right there, and the base is adenine. Down below, we've got a molecule called dideoxyinosidine, sold under the trade name Videx. And the base is weird. The base is not quite the same as adenine. It's called inosine. And the sugar right here doesn't have any oxygens on it. This is called dideoxyribose, which means it can't make a link to other nucleotides. So if a cell is having to make a lot of DNA or RNA, and you fill it full of Videx, the cell will try to use these nucleotides, but as soon as it does, transcription or DNA replication stops, because that oxygen right there is what attaches to the phosphate on the next nucleotide. And if it's missing right there, then a cell can't make DNA or RNA any farther. It shuts down DNA replication or RNA transcription. So if you feed this to a cell that's copying virus DNA or virus RNA, you block those strands from extending any farther. And Videx is one of the cocktails of drugs that's used to treat HIV. And the HIV cocktail consists of these fake nucleotides like Videx, other drugs that interfere with fusion proteins, others that block the virus from assembling, others that block new viruses from breaking out of the host cell. If you've got HIV, it's manageable, but you've got to take so many pills you rattle, because the best strategy is to attack the viruses in multiple ways at the same time. And all of these strategies can be used to treat other viruses, and they're all used now in the treatments that we've developed for coronavirus. And I will stop there and end the recording.